Good day, this is Professor Will, CBMD, PhD. Today's Thursday, June 13, 2019. It is 2:49:59 seconds Eastern Standard Time. And this is a continuation of my scientific research, so let's get started here. How multi-celled animals developed evolutionary discovery rewrite textbooks. Date June 12, 2019. Source University of Queensland. Summary: new findings challenge the long-standing idea that multi-celled animals evolved from a single-celled ancestor resembling a modern sponge cell known as a coenocyte. Using new technology to investigate how multicellular animals developed, their findings revealed a surprising truth. Researchers found that the first multicellular animals probably weren't like the modern-day sponge cells, but were more like a collection of convertible cells. The great-great-great-grandmother of all cells in the animal kingdom, so to speak, was probably quite similar to a stem cell. This is somewhat intuitive as compared to plants and fungi. Animals have many more cell types used in very different ways. From neurons to muscles, and cell flexibility have been critical to animal evolutionary from the start. The findings disprove a long-standing idea that multi-cell animals evolved from a single-celled ancestor resembling a modern sponge cell known as a coenocyte. Scattered throughout the history of evolution are major transitions, including the leap from a world of microscopic single cells to a world of multi-celled animals. With multicellularity came incredible complexity, creating the animal, plant, fungi, and algae kingdoms we see today. These large organisms differ from the other more than 99% of biodiversity that can only be seen under a microscope. The research team mapped individual cells, sequencing all the genes expressed, allowing the researchers to compare similar types of cells over time. A researcher said this meant they could tease out the evolutionary history of individual cell types by searching for the signatures of each type. Biologists for decades believed the existing theory was a no-brainer. As sponge coenocytes look so much like single-celled choanoflagellates, the organism considered to be the closest living relatives of the animals. But their transcriptome signatures simply don't match, meaning that these aren't the core building blocks of animal life that we originally thought they were. This technology has been used only for the last few years, but it has helped us finally address an age-old question, discovering something completely contrary to what anyone had ever proposed. Researchers are taking a core theory of evolutionary biology and turning it on its head. Now we have an opportunity to reimagine the steps that gave rise to the first animals, the underlying rules that turned single cells into multicellular animal life. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, June 13, 2019. It's 3:23:59 seconds Eastern Standard Time, and this is a continuation of my scientific research. So let's get started here. Cause: hardening of the arteries, and potential treatment identified. Date June 12, 2019. Source University of Cambridge. Summary: a team of scientists have identified the mechanism behind hardening of the arteries and shown in animal studies that a generic medication normally used to treat acne could be an effective treatment for the condition. Researchers found that a molecule once thought only to exist inside cells for the purpose of repairing DNA is also responsible for hardening of the arteries, which is associated with dementia, heart disease, high blood pressure, and stroke. There is no current treatment for hardening of the arteries, which is caused by buildup of bone-like calcium deposits, stiffening the arteries and restricting blood flow to organs and tissues. Researchers found that poly-ADP ribose, or PAR, a molecule normally associated with DNA repair, also drives the bone-like calcification of arteries. Additionally, using rats with chronic kidney disease, the researchers found that minocycline, a widely prescribed antibiotic often used to treat acne, could treat hardening of the arteries by preventing the buildup of calcium in the circulatory system. The study is the result of more than a decade of fundamental research is published in the Journal of Cell Report. Artery hardening happens to everyone as they age and accelerated in patients on dialysis, where even children develop calcified arteries. But up until now, we haven't known what controls this process and therefore how to treat it. This hardening or biomineralization is essential for the production of bone, but in arteries it underlies a lot of cardiovascular disease and other diseases associated with aging, like dementia. We wanted to find out what triggers the formation of calcium phosphate crystals, and why it seems to be concentrated around the collagen and elastin which makes up much of the artery wall. Research has shown that PAR, normally associated with the repair of DNA inside the cell, can in fact exist outside the cell, and is the engine of bone production. This led the researchers to hypothesize that PAR may also play a role in biomineralization. In addition, PARP1 and PARP2, the dominant PAR, producing enzymes are expressed in response to DNA damage and oxidative stress, processes which are associated with both bone and vascular calcification. Researchers could see signals from bone that they couldn't explain, so they looked for molecules from first principles to figure it out. Researchers have been thinking for years that hardening of the arteries was linked to DNA damage, and that DNA damage is a pathway switched on by many agents, including smoking and lipids. When this pathway is switched on, it derives the pathologies associated with aging. If enough damage is present, the arteries will eventually reflect it. Using NMR spectroscopy, the researchers found that when the cells become stressed and die, they release PAR, which binds very strong to calcium ions. Once released, the PAR starts mopping up calcium into larger droplets which stick onto the components in artery walls that give the artery its elasticity, where they form ordered crystals and solidify, hardening the arteries. Researchers would never have predicted that it was caused by PAR. It was initially an accidental discovery, but they followed it up, and it has led to a potential therapy. Having discovered the links between DNA damage, PAR, bone, and artery calcification, the researchers then looked into a way of blocking this pathway through the use of PARP inhibitor. We had to find an existing molecule that is cheap and safe, otherwise it would be decades before we would get a treatment. If something has already been shown to be safe in humans, the journey to the clinic can be much safer. Working together with Cycle Pharmaceuticals, a Cambridge-based company, the researchers identified 6 known molecules that they thought might inhibit PARP enzymes. Detailed experiments with these showed that the antibiotic minocycline was highly effective in preventing hardening of the artery. It has been 12 years of basic research to get to this point. Researchers set out with absolutely no expectation of finding a potential treatment. There is no treatment currently, and nobody would have believed us if we had said at that point we were going to cure hardening of the artery. The technology has been patented and has been licensed to Cycle Pharmaceuticals by Cambridge Enterprise, the university's commercialization arm. The researchers are hoping to carry out a proof of principle trial in patients in the next 12 to 18 months. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, June 13, 2019. It is 3:58, 13 seconds Eastern Standard Time. And this is a continuation of my scientific research. So let's get started here. Drug delays type 1 diabetes in people at high risk. Immunotherapy slows progression in clinical disease by 2 years or more. Date June 9, 2019. Source NIH, National Institute of Diabetes and Digestive and Kidney Disease. Summary: a treatment affecting the immune system effectively slowed the progression of clinical type 1 diabetes in high-risk individuals, according to new findings. The study is the first to show that clinical type 1 diabetes can be delayed by 2 or more years among people who are at high risk. The study involving treatment with an anti-CD3 monoclonal antibody, tap-lizumimol, was conducted by Type 1 Diabetes TrialNet, an international collaboration aimed at discovering ways to delay or prevent type 1 diabetes. Researchers enrolled 76 participants ages 8 to 49 who were relatives of people with type 1 diabetes, had at least 2 types of diabetes related to autoantibodies proteins made by the immune system, and abnormal glucose sugar tolerance. Participants were randomly assigned to either the treatment group, which received a 14-day course of tap-lizumimol, or the control group, which received a placebo. All participants received glucose tolerance tests regularly until the study was completed, or until they developed clinical type 1 diabetes, whichever came first. During the trial, 72% of people in the control group developed clinical diabetes, compared to only 43% of the tap-lizumimol group. The median time for people in the control group to develop clinical diabetes was just over 24 months, while those who developed the clinical diabetes in the treatment group had a median time of 48 months before progressing to diagnosis. The difference in outcomes was striking. The discovery and the first evidence we've seen that clinical type 1 diabetes can be delayed with early preventive treatment. The results have important implications for people, particularly youth, who have relatives with the disease, as these individuals may be at high risk and benefit from early screening and treatment. Type 1 diabetes develops when the immune system T cells mistakenly destroy the body's own insulin-producing beta cells. Insulin is needed to convert glucose into energy. Tap-lizumimol targets T cells to lessen the destruction of beta cells. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, June 13, 2019. It is 4:16, 39 seconds Eastern Standard Time. This concludes my scientific reading. Hope you enjoyed listening. Have a great day.